So I think we'll get started. So Eric received his graduate degree in biochemistry and structural biology from Weill Cornell in 1999. This was followed by postdoctoral work in microbial pathogenesis at Yale. Eric's been with us since 2001, and he's currently head of the Laboratory of Structural Microbiology. Eric's received a number of honors, including the Young Investigator Award from the American Society of Microbiology and also the prestigious Eureka Award from the National Institute of Health. His talk is entitled Themes in Host Pathogen Interactions, Insight from Structural Biology. Eric.
Thanks, Ohio. Okay, everyone can hear me. Good, the mic's on. I'll start just by giving you the structure of the talk I'm going to give today. And I wanted to begin by putting the context of this talk into the research that we do, the kinds of questions we're asking, so you get a sense of why we're doing the kinds of things we're doing and what we're trying to accomplish. But the bulk of the talk is going to be focused on an overview of the work we've done since we arrived at Rockefeller. And instead of picking a particular project or something recent that we've been doing, what I wanted to do was to look broadly at not specific projects, but at themes and general insights that we've been able to extract from a multitude of different projects into the host pathogen interaction. And this will, of course, be illustrated by a lot of specific examples. And sprinkled throughout this and also at the end will be some work in progress, some preview of coming attractions, if you will. And there's a full docket, and so I'm going to try to get through this quickly. But for those of you who are a bit impatient or a bit married to the digital age, there's a progress bar here that will gradually fill out throughout the talk from red to green, so you don't panic. So we are interested in understanding the biochemical interactions between pathogenic bacteria and eukaryotic cells. And the questions we tend to ask are basically, at a broad level, how have bacteria adapted to their human environments? And the broader context of that is really human beings are just another surface in the biosphere on the planet. And bacteria, just like any other place, have numerous adaptations to extract resources from that environment and to deal with the hazards of that environment. And what happens in many cases is that bacteria will end up controlling many aspects of host biology through specific modulation of eukaryotic cellular processes. And they've evolved a collection of specialized proteins, so-called virulence factors or effectors, in order to achieve those ends. And many of these are actually delivered directly into eukaryotic cells through what have been termed injectosomes, or molecular syringes, if you will. And that's a good place to kind of anchor what I'm going to be telling you about. So these are large organelle-sized structures that span multiple membranes. Obviously, they have to get things through the eukaryotic membrane, but also from inside the bacterium to outside. And in the case of gram-negative bacteria, the primary focus of my work, that means getting through two different biological membranes. So we focus on the substrates of these systems, what they look like, how they interact and modulate host proteins, and also the injectosomes themselves, how they're constructed, how they're regulated, how the machinery in an energy-dependent fashion engages with these and delivers the substrates. This animation kind of gives you an idea of this process in action. This would be a Salmonella bacterium. Here, assembling one of these injectosomes, a so-called type 3 secretion system, and through this, injecting a large number of virulence factors that work together to achieve a biological goal. They manipulate host cell biology, cytoskeletal structure, and so forth. And in the case of Salmonella, achieve a membrane ruffling that will internalize the bacterium in a macropinocytotic event. So when we first came to Rockefeller, amazingly enough, almost 12 years ago now, we set out two goals for ourselves. The first was a little more prosaic, and it was basically, we were just hoping to be able to determine a number of important crystal structures of bacterial virulence factors, either themselves or, more importantly, in complex with host targets, to really understand at the molecular level how bacteria were reprogramming into biology. And then what we wanted to do was take that information and use very targeted structure-based approaches, mutagenesis and so forth, to explore these interactions at the cellular level and, when possible, in an infection biology context. Our second goal would be dependent on the first. If we could determine enough of these, what we hope to be able to do would be to extract more general paradigms of this host-pathogen interaction, and basically into two areas, as I divided the last images into, the substrates, how they modulate the host biology, and also the injectosomes themselves, how they recognize, engage, and deliver these substrates into the host cell. So I'm going to say just a little bit about the first, but it'll be mostly a prelude to the second. We've mostly been quite successful with this. This just summarizes a lot of what's been going on over the last decade or so. And we have now many structures, especially complexes, in many areas of host cell biology in terms of where the pathogen is impacting. And that's been very fruitful both in understanding pathogenesis, and we've also been able to do these things like mutagenize these things, look at how this affects virulence and so forth, and I'll give you examples of that. But also, besides the moving on to the second goal, which I'll get to in a second, I think one thing that's exciting is it provides a library for structure-based drug design, something that's been of a lot of interest recently in both medicinal chemistry and infection biology, is trying to develop inhibitors of bacterial virulence factors. The idea would be if we can develop compounds that will inhibit key virulence proteins, what we may achieve is a therapeutic effect without necessarily having to find a way to target the viability of the organism. And there's been a lot of work on that. And one of the first steps to that would be, at least at one stage of drug development, would be coming up with a lot of structures of important virulence factors. And so we're glad to be able to contribute to that effort. So then, now that we have summarized this, and not really shown you too many examples, but assuming that this is true, taking these examples of structures of bacterial virulence factors and their hosts, how can we develop paradigms, and in the first place, for the interaction of these factors with the eukaryotic cell? And that's what I'm going to focus a good bulk of the talk on. And what I'm going to do is I'm going to divide these paradigms into three themes: mimicry, elegance, and redundancy. And I'll actually spend most of the time on mimicry, but I'll try to give some respect to these two as well. And I'll begin by defining, at least in a general sense, what I mean by mimic. Mimicry or mimics in the biological sciences are used in a lot of very different ways. I mean something fairly simple, which is essentially a bacterial protein that can fall into one of two categories. It possesses either structural and/or sequence similarity to eukaryotic factors, so something that probably shared a common ancestor with the eukaryotic protein, or something that does not have any structural sequence similarity to eukaryotic proteins, but nonetheless, through its molecular surfaces and chemical groups, is able to achieve similar biochemical effects or modulation of the host cell as eukaryotic factors do. And one might potentially think of these as factors that have been convergently evolved. And I'll show you examples of both of these. In fact, what I'm going to do is show you 5 examples of proteins that I think illustrate this theme of mimicry very well. Now, because there are this many examples, I won't have time to go into an enormous amount of detail on each one. But what I hope to do is give enough background biology and focus specifically on the aspects of mimicry to illustrate this theme as best I can. And we'll be jumping between many different pathogens in this process, so hope if there are any confusions or questions, please ask them hopefully at the end of the talk, so I can make sure I finish this all up. So I'll begin with Salmonella, a protein called CYP-A, and this was in fact the first project we worked on when we came to Rockefeller. And it's part of this program that you might remember from the little animation I showed you, in which Salmonella induces its own uptake into cells by reprogramming cytoskeletal structure. Now, Salmonella does this, as I mentioned, through many factors, some of which indirectly reorganize the actin cytoskeleton by, for example, interacting with signal transduction molecules like the Rho-GTPases. But it also directly does so through binding to cytoskeletal proteins themselves, such as actin. And that's where CYP-A comes in. So CYP-A is shown schematically here. And I'm going to spend just a second here describing the modular structure of this, because it's going to come up again and again throughout this talk. So many of the substrates of the type 3 secretion injectosome can follow into this sort of a pattern. You have at the N-terminus a secretion peptide, somewhere between 20 and 25 amino acids, followed immediately by a so-called translocation domain. And this interacts with specific so-called secretion chaperones in the bacterium. Both of these are critical for the proper engagement with the injectosome and delivery into the host cell. So once you move beyond this delivery section, you have one or more domains that function within the host. And CypA is characterized at the C-terminus by a domain that polymerizes actin. It can reduce the critical concentration required to form actin filaments. And as you see here, it's very important in an in vivo context. So this is just a video that shows some dramatic membrane ruffling when cells are exposed to salmonella. And in the still, the actin cytoskeleton is shown in red and the bacteria in yellow. And what you can see is wild-type salmonella induces these very pronounced, localized cytoskeletal alterations. However, when you knock out CypA, what you find is that you've made these much less robust, more diffuse. And in fact, CypA mutants have an internalization defect. They delay the process significantly. And so this ability to polymerize actin by this protein is quite important. So we combined X-ray crystallography and electron microscopy to try to understand how CypA was functioning, and we came up with the following model. In the EM, you can see the actin filament modeled in red, and then that filament is studded on the outside with these yellow blobs. If you move over here, these yellow blobs turn out to be this crystallized domain of CypA. We've modeled it in here. And the termini rest right alongside this extra density that we don't have in our crystal structure. This is disordered bits, which we've called arms. And what you see in the actin filament is that these arms are connecting different protomers in the filament. And this led us to a model of how this was functioning, which we termed a molecular staple. And we did a lot of things to verify that by truncating the arms and so forth and showing that they were important for the polymerization activity. But I won't go into that. Well, one other thing is that if you extend the polypeptide from either end, you increase the length and the size of these arms, but maintain the globular section. But what I wanted to focus on in this theme of mimicry is how CypA is acting like a host protein. So there's a protein called nebulin, which is present at high levels in muscle cells, which achieves something very similar. So if you look at the actin filament here, you can see these bridging staples, if you will, that are nebulin. But nebulin would fit into, CypA would fit into this category of mimics that don't look like the host but achieve something very similar. Because you see it's got this globular domain with arms, whereas nebulin doesn't contain a globular domain, but is a long alpha helical molecule. So here's a protein that, while it doesn't look very much like the host protein, can achieve something very similar in this molecular staple function to achieve the same goal, which is enhancing actin polymerization. So the second example, I'll switch to the pathogen Yersinia and a protein called YPKA. Now, some background on Yersinia. It's a gram-negative pathogen as well. There are 3 pathogenic species, Enterocolitica, pseudotuberculosis, and the more infamous Yersinia pestis, which is responsible for very well-known pandemics historically that probably have killed, at least estimates are on the order of 200 million people in the past, upwards of maybe 50% of some population centers. Yersinia, like Salmonella, uses a protein injectosome, a type 3 secretion system, and one of the proteins that goes through this, the substrate, is Yersinia protein kinase A, or YPKA. It's an essential virulence factor that on its own has a very dramatic effect on cells. Here's a cell that's been transfected with YPKA, and you can see the normally robust actin cytoskeleton in the nearby cells is gone, and in fact these cells will shrivel up and detach and die. Here's a schematic to show you the organization of this protein. Again, the secretion peptide, the translocation domain binding a chaperone, important for delivery. And then YPKA has 2 functional subunits. One has sequence similarity to eukaryotic serine-threonine kinases, and therefore would fit into this mimic, the first category. And it has a known target now, G-alpha-Q protein. And the C-terminal domain, which at the time we began work on this, it was not clear what it was doing outside of the fact that it interacted with the Rho GTPases. Summarize a lot of work to get to the chase. YPKA interacts with the Rho GTPases, RAC1 in this case, in our structure, at the key signaling elements of this molecule, the switch 1 and switch 2 regions, shown right here. And even more interestingly, when we compared this complex to the database of structures, what we found is that the interaction between YPKA and RAC1 very closely resembled a subset of what was in the PDB, the interaction of so-called Rho GDIs with their small GTPases. Not only in certain contacts, like these acidic residues which fall on top of each other between these different proteins, but more significantly in the conformation information of the key regulatory regions of the GTPases, such as switch 1, which were almost identical. And this suggested that YPK may be mimicking these proteins, these Rho-GDIs. And that's what we set out to try to show. And let me try to put this in the context of this pathway so you'll understand the significance, if you don't already. The Rho-GTPases are on-off switches, depending on whether they're binding GTP, in which case the switch 1 and switch 2 regions are in conformations where they can bind effectors that transduce signals, many of those involved in reorganizing the cytoskeleton of the cell. But if this is hydrolyzed to GDP, often accelerated by GTPase activating proteins, those two regions change conformation, and this goes into a signal off state, and they can no longer bind and transduce these signals. The GDIs do something in addition to shunt this to off. They do two things. The first is they bind the lipidated tail, which anchors these in the membrane, removing them from the membrane, where they have to be active. And the second thing they do is they prevent nucleotide exchange, so you cannot activate these proteins anymore. So we were able to show, and I'm not going to go through all the data, that YPK only could do one of these things. It couldn't bind the C-terminal tail, but it was quite efficient at inhibiting nucleotide exchange, even in the context of factors that would promote it. And we were able, because we had the structure, to make very targeted mutagenesis that did not disrupt the fold of the proteins, but which was able to destabilize the complex. And when we did that, YPK would lose its ability to inhibit this nucleotide exchange. We were able to then take that both into cell biology and show that this loss of GDI activity in the test tube actually correlated to an inability to muck around with the cytoskeleton. And even more importantly, we could carry it all the way to a virulence phenotype in a mouse infection. So here is shown a wild type and a single loss of contact mutation in YPKA between YPK and the Rho-GTPases. And what you see is a huge difference in virulence. The mice infected with the wild type are mostly dead in a week, whereas those infected with this mutant most survive, and even those that die, it takes them longer than the wild type. So a very pronounced effect. So this was a very exciting project because we were able to not only perform a structure-based discovery program to understand what this effector was doing, but it revealed that this was a very fascinating mimic of a eukaryotic regulator, and that this activity was absolutely critical to virulence in an infection. The third example, I'm going to switch to the pathogen Helicobacter pylori and a protein called CAG-A. So Helicobacter pylori, as many of you probably know, it's also a gram-negative pathogen. 50% at present of the human population is infected, 1 out of every 2 people. And if you look at this geographical distribution, what you can conclude very quickly is that this number is probably historically very low, and it's mainly in areas of very high antibiotic use. So historically, probably 80 to 90% of the population has been infected with this organism. It colonizes the stomach. In fact, it's the only known bacterium that is able to do so. But in most cases, it's asymptomatic. And only in a few percent does it cause gastritis and peptic ulceration. And it's because of this that it is actually the most significant risk factor for gastric adenocarcinoma in the world, leading the World Health Organization to classify the organism itself as a carcinogen. So the CAG-A protein of Helicobacter is an important virulence factor, and it's translocated into host cells by one of these injectosomes, in this case a type 4 secretion system, which is structurally distinct from the type 3 secretion systems, but functionally isomorphous, if you will. CAG-A is a rather large protein, around 1,200 amino acids. It interacts with numerous cellular proteins. Somewhere between 10 and 15 have been identified and continue to be identified. It increases virulence. It's associated with an increased risk of cancer, and in fact, CAG-A is tumorigenic in and of itself in animal models. And it induces many changes in the cell, visibly cytoskeletal alterations, as well as numerous changes in signal transduction because of all the proteins it interacts with. So we got into this project looking at a C-terminal region of CAG-A that was known to interact and inhibit a protein called MARC2. And let me give you the background on this. MARC2 is a serine threonine kinase in the host cell, a very important one because it is a key regulator of cell polarity and tight junctions. It's part of these so-called polarity kinases. Helicobacter pylori, and here's where the infection side comes in, is unable to grow on the apical side of polarized cells without CAG-A. However, the CAG-A-induced depolarization, in part due to its inhibition of MARC2, renders the apical side permissive for growth. So you see a strong mandate for the use of CAG-A to inhibit this kinase. What I'm showing here, just spinning around, is a complex of the kinase in blue with a peptide from CAG-A that is absolutely sufficient for inhibiting the activity of the kinase. And where the interesting story in mimicry comes in, I've tried to illustrate here. So what I've done is I've shown the molecular surface of the kinase, kind of in gray for the most part, and I've also aligned another eukaryotic complex with a kinase, the protein kinase A with its inhibitor PKI here in green. And the reason I do that is to illustrate the similarities between how the CAG-A peptide is binding its kinase and how PKI inhibits PKA. They're both peptide inhibitors, short regions that sit actually in the substrate binding site of the kinase. Right here is where the substrate would bind, making it impossible for substrates to be phosphorylated. This is the ATP where gamma phosphate would be transferred. Not only does it have sort of a three-space similarity, there are even some contacts, amazingly enough, that are apparently similar, I wouldn't call them conserved, between these two inhibitors. There's an arginine, which interacts with an aspartic acid in both these proteins, even though the kinases aren't necessarily that closely related, and several hydrophobic contacts that bind in similar pockets. Where you really see the mimicry, though, is when you look at the sequence level. So what I've done here is I've aligned a Western and East Asian strain of CAG-A, just that region, the peptide, and aligned with that about 10 or so sequences where here's the serine that would be phosphorylated. These are the substrate regions in the substrate family for MARC2. And these starred residues with these red boxes are 4 residues that kind of plug into the kinase from CAG-A, the key interaction regions. And what you can see is that there's a high level of similarity between the residues CAG-A has here and what you see conserved in the substrate family. So not only is CAG-A interacting in the substrate binding site like a eukaryotic inhibitor, but it's even mimicking the substrate family in terms of how it recognizes MARC2. So it's a really amazing example of mimicking host proteins in order to achieve this biological result in depolarizing the cells. Here's the first example of a little bit of work in progress I wanted to give you because it's something we're very excited about. We've been able to identify structural subdomains in the N-terminus of CAG-A that are very stable and well-behaved, and we've used those as bait in the yeast-2 hybrid analysis. And to cut to the chase, our top hit was a protein called ASP2, which has us very excited. The reason being is that ASP2 is a member of a family of proteins that is pro-apoptotic and that are classified as tumor suppressors. These are proteins that interact with other important tumor suppressors, like the p53 family, for example, and drive the apoptotic pathway in response to a variety of signals. And so what we have then, sort of in a summary, is we have what's been termed a bacterial oncoprotein, CAG-A, which we now have identified as interacting with a host tumor suppressor, ASP2. And so not only is this potentially interesting in terms of the infection biology, why is this happening in terms of what CAG-A is trying to accomplish, but the other question will be, will this be able to potentially give us some insights into the tumorigenic nature of this protein and this pathogen? We've been able to make some progress with this. We have stable biochemical complexes that we can purify at high level, been able to crystallize these, and very recently we determined the structure of a CAG-A ASP2 complex. And I won't go into all the details of that here. This is something that we're actively pursuing right now, except to say that what this has allowed us to do is to make a library of mutations that very specific structure-based mutants that don't disrupt these folds, but destabilize the complex. And we're now taking those mutants and trying to use those as probes or tools in cell biological and infection biological assays to try to get at both the infection biology and potentially the deregulation of pathways in the host cell that might be oncogenic. Okay, the fourth example of mimicry I want to give you, we're going to take an even bigger leap out of the stomach and into plant pathogens. And this is from Pseudomonas syringae, and it's a protein called AVR-PTOB. And to understand this story, I need to back up and tell you a little bit about the really amazing immune system, so to speak, that plants have to deal with bacterial pathogens. One of the key resistances to bacterial infection that many plants have is a protein recognition system in which these type 3 secretion systems that inject proteins to manipulate the plant cells, just like they do animal cells, the plants have evolved a way to recognize some of these and initiate signal transduction, which leads to a scorched earth policy in effect. It's called the hypersensitive response, and a key element of that is programmed cell death, localized. So you can see these splotches here. So what happens is the plant essentially trashes the region that it detects infection by the bacterium, sparing the rest of the plant from a systemic infection, and it just confers immunity in many cases. But of course, that leads bacteria to have selective pressure to evolve strategies to counteract this immunity-associated programmed cell death. And that's where this story comes in with AVR-PTOB. It's a protein that the C-terminus is sufficient in and of itself to suppress programmed cell death in plants. It's an anti-PCD domain. So that's what got us interested in this, and we determined the crystal structure of a C-terminal domain that possesses activity. And I'll cut to the chase on that because we were very surprised to find the following, is that it had both structural as well as functional homology to a set of proteins in eukaryotic cells called ring finger U-box proteins. So this just shows the backbone alignment with AVR-PTOB and several host ring fingers and U-boxes, as well as a patch of amino acids is critical to the functioning of these in eukaryotic cells. And that's what I'll explain to you what this function is, if you don't know. So the ring finger U-box proteins are part of a family of proteins that function in the ubiquitination pathways in eukaryotic cells. They're part of the ligases that transfer ubiquitin to substrates. And in particular, what these proteins do is they bind what are called E2 proteins that are conjugated to ubiquitin. So essentially, they're adapters that bring in ubiquitin to a multi-protein ligase complex, highly regulated process. AVR-PTOB had this very strong homology to these proteins. And in fact, what we could show is that AVR-PTOB could function as an E3 ligase in the test tube, and that mutating those key residues that are involved in adapting, bringing in ubiquitin, will kill the ligase activity. Even more interestingly, we're able to take those same mutants, single point mutants, and show that not only did they abrogate ligase activity, but the protein lost its anti-PCD activity when injected into plant cells. Even more interestingly, we could carry the ligase activity connected to the programmed cell death activity and connect that loss in the mutants to a reduction in virulence. When we infected plants with wild type or mutant, the mutant bacteria were several logs less after we recovered from the plants in terms of how much they had replicated. So here we have a really amazing example of the first type of mimicry. It appears to be very structurally and even has sequence similarity to host factors, probably shares a common ancestor, and that mimicry gives it a very important activity that is critical for virulence. In case you're wondering what in the world this ligase activity is doing in the plant cell, it's ubiquitinating some key signal transduction kinases that are part of the programmed cell death pathway in response to these proteins. And so it shuts that down and the plant can't mount this immune response. And finally, the fifth example I wanted to give in the theme of mimicry is back to Salmonella and a protein called SHPH2. But we'll be staying in the area of E3 ligases. SHPH2 is an important protein not for the internalization of Salmonella, but for its ability to spread systemically throughout the organism during infection. And we were able to show that SHPH2, like AVR-PTOB, is an E3 ubiquitin ligase. We were able to show that in vitro, and there was a critical cysteine residue that was the catalytic residue for this process. In that work, we were able to crystallize this protein. It consists of a leucine-rich repeat domain and a C-terminal domain, here shown in red, which harbors the entire ligase activity as well as the catalytic cysteine. If you remove the leucine-rich repeat domain, this domain is active as a ligase. And in fact, it's essentially auto-regulated. The leucine-rich repeat domain in our structure, which you can probably see a little bit better here, is sequestered by is sequestering the cysteine. So there's really, it's hard to imagine how you'd be able to get a ubiquitin ligation reaction going on here with substrate and other proteins. However, at the same time that we solved the structure from Salmonella, there's a structure of a homolog from Shigella, another pathogen, which crystallizes in a different conformation. So we had hypothesized that this domain would have to undergo a conformational change, and that's exactly what you see in this homologous structure. They managed to crystallize it in an open conformation where the C-terminal domain has flipped out 180 degrees, exposing the active site cysteine. So that was interesting. We were able to come up with some models for the biochemical activity of this domain. But what I really wanted to do, and what's fun in this theme of mimicry, is to put it in the context of all of these mimics of E3 ubiquitin ligation from bacteria. So in eukaryotic cells, there are basically two classes of ligase, E3 ligase. There are the ring finger proteins I told you about, which bacteria have a mimic, and I showed you that. But there's also the so-called HEC domains. These use a catalytic cysteine as well. And it turns out bacteria also have a mimic of this. And I didn't really show you this. This is a protein called SOP-A from Salmonella. But there's very good structural homology to this family as well. So bacteria have covered, so to speak, both bases in terms of the types of E3 ubiquitin ligases in the host cell. But to top it all off, to give us the hat trick, we have a new domain, which the NEL domain that I just showed you from SHPH2 for novel E3 ligase, that's what we called it, which doesn't look like anything in the host, but still achieves the same biochemical activity, which is the transfer of ubiquitin. So both of mimics of host factors, as well as something that looks like a convergently evolved mimic as well, to throw it all in. Okay, so that was the bulk of sort of the paradigms on the substrates of these injectosomes. I do want to say a little bit on the other two themes. And I could start off by just saying any of those examples, in my opinion anyway, would serve to illustrate this idea of elegance. And I use this theme here because I think until recently, while it was appreciated that viruses could be very sophisticated and elegant in how they manipulated eukaryotic cells, it wasn't as appreciated that bacteria could also be. They were often thought of as manipulating things through toxins that caused really disastrous results that didn't seem very elegant. But most of the examples I was showing you are interesting, very precise, in many cases reversible modifications in the host cell. But I want to give you one more example. And this is a protein called cycle inhibiting factor, CIF. It's found in many gram-negative pathogens, particularly E. coli species. And it's called this because it is able to inhibit cell cycle progression in eukaryotic cells, which is why we began working on this. That was a fascinating activity. You see many hallmarks of arrested cells in the presence of CIF, such as hyperphosphorylated CDK1, the accumulation of cell cycle inhibitors like P21 and P27, and so forth. And we determined the first structure of one of these family members from E. coli, and we found that it defined a novel clan of the cysteine protease superfamily. And it's a poorly named superfamily because it spans a large number of very different and interesting enzymatic activities. Everything from proteolysis to acetyltransferases, transglutamases, deamidases. They all are part of this family and share a core structure, which is a beta sheet, shown in orange here with several different ones, an alpha helix, and in particular, these elements that present a catalytic triad that's conserved in all of them. In particular, there's a cysteine and a histidine, which is invariant, and then a third residue, which until the structure of CIF came out, was thought to always be either an aspartic acid or an asparagine. But in the case of CIF, turned out to be a glutamine. But that didn't tell us what CIF did, for the very reason I told you this family of proteins can share a common catalytic core, but can do many different things. To try to get at the function, we again pursued a genetics approach. We did a UST hybrid screen with what we hoped to be a substrate trap. Cysteine to alanine mutation in CIF, which was inactive and actually did work. With this mutant, but not with the wild type, we were able to pull out a protein called NET8. And this was a very exciting discovery, and if you know about cell cycle regulation, that probably makes sense to you, and I'll try to explain it here. So NET8 is a small ubiquitin-like protein, like the protein SUMO. They have very high homology to ubiquitin, but they tend to be put on like monoubiquitination for regulatory purposes. And in fact, NET8 is conjugated to a class of proteins called cullins. Cullins, and here we come right back to the E3 ligases again, are subunits of these large ring finger-based E3 ligases, schematically shown here. And in particular, Col1 is associated with a protein complex called the SCF complex, which is an E3 ligase that is critical to the G1 to S transition in the progression through the cell cycle. In particular, it ubiquitinates two cell cycle inhibitors, P21 and more importantly in this case P27, which inhibit cyclin-dependent kinases that are important for progression of the cell cycle. NET8 is not only conjugated to COL1, but the proper conjugation of NET8 to COL1 is critical for this complex to be active. And that's where we see the connection with CYF and the rest of the cell cycle. It turns out that CYF is a deamidase that targets glutamine 40 of NET8. And this deamidation inhibits the ability of NET8 to activate the SCF complex. What that results in is a dead E3 ligase in which the cell cycle inhibitors are not targeted for degradation. It stay at high levels in the cell, preventing progression through the cell cycle. And the reason I've used this as an illustration of elegance is basically you have a bacterial virulence factor that is able to make a single atomic mass unit change in the cell and arrest cell division. And I just find that amazing and beautiful and very sophisticated considering the context in which this is operating. Finally, in terms of the paradigms, I want to just mention briefly redundancy. This, you probably can already guess, is something that is easily illustrated in the process of ubiquitination. Something that's very specific to eukaryotic cells, has several different points in which ubiquitin is cycled from an energy-dependent conjugation to E1s, a handoff to E2s, the large regulated E3 ligases that bring in substrates, and finally the removal of ubiquitin and recycling of the protein. It turns out that I've shown you several examples of ligases, but bacteria target every single one of these steps, from the initial conjugation all the way to having deubiquitinases that remove ubiquitin from proteins. So in that sense, bacteria are redundantly targeting every single aspect of this process in the eukaryotic cell. I wanted to go back for a second example and look again at Yersinia and its effect on the Rho-GTPase signaling pathway. And this is critical in the reorganization of the actin cytoskeleton. And that's critical to the functioning of professional phagocytes. And that's why it's important to Yersinia. Yersinia shunts this entire pathway to off. It does so in with three key virulence factors that are delivered by an injectosome. YAP-E, which turns out to be a gap stimulating GTP hydrolysis, turning this off. It has a protein called YAP-T, which is a bona fide cysteine protease, which cleaves the C-terminal tails of these proteins, removing them from the membrane so they won't be in the right place to engage with the signal transduction machinery. And finally, as I mentioned earlier, there's this GDI mimic, YPKA, which prevents nucleotide exchange in these proteins, further ensuring that this pathway is off. And therefore, what you see when Yersinia injects its battery of virulence factors is that macrophages, for example, are completely paralyzed and can't internalize the pathogen. And finally, back to Salmonella for redundancy. I mentioned early on that there were several proteins that are involved in achieving these membrane ruffles that internalize the pathogen. I talked about CYP-A, which helps to polymerize actin. There are other proteins that bind actin and nucleate it. There are proteins that indirectly, in signal transduction cascades, stimulate cytoskeletal rearrangements, including a GEF of the Rho-GTPases. And then to return once again to that animation, I can put all of this in context and say that when Salmonella uses its injectosome to inject all of these different proteins that are impinging in different ways upon the cytoskeleton, we have a great illustration of this theme of redundancy by bacterial pathogens, including here CYP-A, which is a stapling filament leading to polymerization underneath the pathogen and subsequent internalization, where Salmonella will replicate. Okay, so that's the bulk, 2/3 of the talk, let's say, where I focused on the substrates. And I tried to show you how the structural biology that we've done over the last 12 years has allowed us to elicit or characterize these different themes or paradigms in the biology in the host pathogen interaction. So I want to switch from the substrates now, and in the last portion of the talk about the injectosomes themselves. And I've divided this into the following somewhat arbitrary categories of recognition, engagement, which is really also part of recognition, and then the delivery. And so obviously proteins have to be recognized, selected from the thousands of proteins that are in the bacterium for secretion. They have to be properly bound and engaged and with the secretion apparatus itself, and I'll go into some of this. And finally, in an energy-dependent fashion, the machinery has to force these proteins through the large secretion channel and into the eukaryotic cell while they're functioning. And the context of this is that gram-negative bacteria really have a significant topology problem. Not only do they need to get things through the eukaryotic membrane, but they have to get them through both their inner and outer membranes. So there's a lot that they have to solve, but they've been pretty good at it. There are now 6 genetically and functionally distinct known protein secretion systems, and the one we're focusing on, again, is called the type 3 secretion system. So in the beginning, in terms of selecting what proteins are going to go through these systems, I'll return to the sort of diagram I showed you earlier. So there are 2 key elements in the recognition and proper targeting of these proteins. The first is the secretion peptide, and the second is the so-called translocation domain, and I'll talk about each of these in sequence. The secretion peptide, amazingly enough, it's the first thing that's really was identified. It's the first 20 to 25 amino acids of these substrates. And it's critical. If you remove it, these proteins are not secreted. If you slap this onto a heterologous protein, in many cases those will be secreted. However, we still don't understand the nature of the signal. We have been unable to determine that in 15 years of work, which is kind of astounding in a way. And there's been a lot of debate, sometimes bordering on not friendly. What we've managed to do is, the best I would say, is develop machine learning algorithms for recognizing what is going to be a substrate and what isn't. What they do is they train these learning algorithms on a subset of effectors, and we have hundreds and hundreds of these that are known now, so there's a decent database. And then they learn how to recognize something in the secretion peptide, and then can predict to decent accuracy whether a protein will or will not be a substrate for these systems. However, these machine learning algorithms don't really give a very good human insight into what the signal is, so we're still working on that. The second portion, the translocation domain, we've made a lot more progress, and that's something we've been very involved in. And so, actually, one of the first, well, let me tell you a little bit about the translocation domain. It binds, as I mentioned, these secretion chaperones. And if you either delete the translocation domain or knock out the chaperone, what you will find is, in many cases, an interesting phenotype. The protein, if it's not just degraded and remains within the bacterium, will not get into the host cell. It can't be translocated. However, in many cases, they can be secreted. And in several cases, it's been shown that the manner in which these are secreted is not through the pathogenic type 3 secretion system, but through an evolutionary cousin, which is the flagellar type 3 secretion system. So type 3 secretion systems are not only used for pathogenesis, but they're used in many different areas where you need to get proteins through membranes, including motility and the flagellum, as well as symbiotic relationships. But that tells you something interesting, which is that 2 things, I think. Anyway, the first is something about the secretion peptide, which is that it must be old enough, whatever the information is in this peptide, that it's still conserved enough that it can be, in many cases, recognized by the flagellar system. And the other thing it tells you is that the translocation domain is serving, in these cases, as a very specific targeting signal to get it to the right type 3 secretion system. So this was one of the first things that we made an impact on in the field. In fact, we saw the first structure of a secretion chaperone substrate complex, which is shown spinning around here, which was pretty interesting because the chaperones are shown in these gray blobs, nice globular proteins, but the substrates, the chaperone binding domain here, translocation domain, was wrapped around the proteins in a distinctly non-globular form, which led us to propose a model that this was priming these in a secretion-competent state for translocation. And the reason we propose that is that the channel in the injectosomes, it's on the order of 25 angstroms, is not big enough to allow most globular proteins through. And it's been thought for a long time that proteins had to go in a non-globular state. But since this initial structure, we've been able to solve a number of these in complex with their substrates. And this was another case where, given a large number of protein structures, we were able to abstract from the structures a more general principle, which was a signal in this domain, which we've termed the beta motif. I can sort of show it more simply here. Here is, again, the CYP-A protein, its translocation domain, interacting with its secretion chaperone INF-B. And what you can see are there are 3 amino acids that plug into a hydrophobic pocket in the chaperone. And the reason it's called the beta motif is this region here, and all the chaperone substrate complexes that have been determined, forms an intermolecular beta sheet between the chaperone and the substrate. And these 3 residues come off that beta strand that bridges the 2 proteins. And you can see that here I've aligned a large number of these with a generic chaperone. They all come through the same region right here, making that intermolecular beta sheet. Here's the strand itself, where all the residues fall on top of each other, essentially. And here is the pocket and the chaperones aligned from numerous chaperones in which this motif plugs. So this has been, in my opinion, a very important discovery, one in which we finally have been able to extract a signal in type 3 secretion, and one which could not have been found by analyzing the sequences. This was really only evident after we had numerous protein structures in hand and could compare them and align them. And we recently were able to actually extend this from the animal pathogens all the way to the plants. So what it looks like is that this beta motif signal is highly conserved, probably a very old signal as well, and is found at present in every single complex of this type that's been done from animals to plants. But that just made us more interested to understand what the far N-terminal peptide would be, and in particular gave us an idea that maybe structural biology might be the way to go after it. And so what we hypothesize, and this is a project that we've been working on now for almost half a decade, is if we could, because the problem is in these chaperone substrate complexes, that N-terminal peptide is disordered. The chaperone does not interact with that. The question is what actually does, and we're not sure. But we know that there are a number of proteins that assemble. This would be the injectosome here, including the integral membrane proteins. But there are several, both cytoplasmic domains of these and other proteins that come in and form a very large macromolecular machinery that regulates this process, including an ATPase, which is critical for the process. What we hoped was if we could put together some of these higher order complexes, we would achieve one of them that was stable, and also the recognition event would occur in that complex. The problem, and why we've been spending half a decade on this, is that it has been extremely difficult to put together any sort of stable complexes here, which was very surprising. We didn't anticipate that. As a work in progress, something I can say is that finally, one of the things we've been able to do is make some higher order complexes. One I'm showing here is a ternate complex with the ATPase and two other proteins that we can purify stably over gel filtration, for example, and also a couple binary complexes that are interesting. And what we hope is not only to study this, to understand aspects of how this machinery is organized, but we're hoping to be able to bring in the chaperone substrate complexes and form a higher order complex, and finally get at how this peptide is recognized. And hopefully, like in the case with the translocation domains, we'll be able to extract what the signal is from the protein structures. At least that's our goal. Finally, the last part is of obvious interest is the channel itself. And this is an area that's had probably some of the most progress on the injectosomes. Back in 1998, the first images, negative stain EM, were obtained of this device. Since then, cryo-EM is pretty much taken over and pushed the resolution somewhere in the range of 5 to 10 angstroms, depending on who's doing it and what region of the apparatus you're looking at. The structure itself, interestingly, is, for the bulk, composed of only 3 proteins, which, because of its size, tells you that these are going to be high-order oligomers. Here are the proteins themselves. In Salmonella, it's Mg, PERG-H, and PERG-K. This would be the outer membrane, the inner membrane, the periplasmic space. EM tends to agree that the inner rings are on the order of 24-fold symmetric. There's some debate on the symmetry of the outer ring, which includes this neck region. Different groups, depending on who's analyzing it and the techniques they use, they get somewhere between 12 to 15-fold symmetry, which puts the size of this apparatus, this channel, on the order of 2.5 to 3.5 megadaltons. Now, there has been some progress in high-resolution structures of this, but they've mainly been either cytoplasmic or periplasmic subdomains of these proteins. But there have been some interesting insights, not so much in how this is organized. There are competing models for how these proteins might be packing into some of this density. But one thing I think that is very interesting that came out of this is an analysis we did from one of the structures that we solved, and in comparison to several others from different elements of the proteins here, we found that they all fell into similar classes, in that there were 2 primary classes that shared the basic same fold. So what we had was a model in which we proposed that the type 3 secretion channel, and even some of the components in the engine, emerged as a series of gene duplication events. So these different kinds of Legos packing together to create this apparatus. But like I said, it really didn't tell us how it was put together. And so what we believe is we need to do high-resolution X-ray crystallography of the assembled type 3 secretion system itself. And this is a difficult challenge. There's a size problem. For comparison, here's the ribosome, here's the injectosome. Yes, it's on the order of the size of the ribosome, a little bit larger, but there are other complications that make it a little bit harder. The ribosome, for example, is produced at high levels in many different cells. The injectosome is not. It can't be. It's toxic. Puts these large pores into the membrane. So the ability to isolate a lot of this is challenging. The ribosome exists in a lot of thermophilic organisms, which made crystallography, accelerated the process of X-ray crystallography on this machine. As far as we know, there are no thermophilic organisms with type 3 secretion systems. And finally, the ribosome is a soluble protein, whereas this is a dual membrane spanning channel. So we've got a lot of challenges in front of us. Nonetheless, over the last 3 years we've been working on this, we've had some proof of principle that have us very excited. We are able to purify this and even crystallize it, only diffracting to about a little beyond 10 angstroms. But nonetheless, that was very exciting. These crystals contain the constituent elements of the type 3 secretion system. And it gave us the idea, the belief that this is a project that is doable over the long term. So that's what I want to say to you today. I'm sorry it was a full docket, like I said. Just to summarize, I tried to show you that using structural biology, particularly when you have a large library of structures, it gives you the opportunity to develop general paradigms about your biological systems. And I tried to show you some ideas I've had about what you can extract from the structures that we've worked on. And even we see that is the case in understanding the injectosomes themselves and not just the substrates, and in particular the translocation domain signal and the beta motif that I told you was directly the product of structural analysis and really couldn't have come any other way. And that's basically, in terms of what we're doing for the future, more of the same. We're studying the virulence factors, complexes with host targets, and trying to understand the functioning of the injectosome. And so then, summarizing 12 years, it's really impossible to thank everybody specifically, but I'll try to give some overall acknowledgments. People from the lab, there are many that aren't listed here, and I apologize for that. These are directly related to the projects that I talked about today. There are many people at Rockefeller, heads of lab, but also heads of departments who have either collaborated with us or have been extremely helpful in sharing equipment and resources and advice, and I want to thank them. And yeah, this is fun. And finally, obviously we had a lot of collaborators, particularly on some of the organismal infection models, and we're very grateful to their efforts and their working with them. And finally, the last thing I wanted to do was acknowledge the broader context of Rockefeller, because we're usually doing experiments and not thinking about anything else except our experiments. And the reason we can do that is because of some of the people I'm listing here, these departments. There are so many people at the university that are working very hard, communications, finance, development, the dean's office, on, and I want to acknowledge them because without them, we wouldn't be able to do the things that we do. And so you can call me a Kenyan terrorist socialist, but I believe it takes a village, and I do want to acknowledge everyone's assistance in letting us do our science. So thank you. Questions? Complaints?
This beta motif you found, you said, and I know that the signal vector, some of them are sufficient.
Right. Right.
Is it known that, for example, in the cases where the signal vector is not sufficient, that this beta motif is missing?
Right, yeah, so no, in the cases where you don't have, there are different ways you could target the substrates to the type 3 secretion system. The chaperone effector complexes are one broad method of doing that. And in all of those to date where you have a chaperone effector interaction, there's been a beta motif. But they're not the only ways. In fact, if you look at one, Salmonella, for example, has 2 different pathogenicity islands. And a lot of the substrates that are involved in the second pathogenicity island's unique type 3 secretion system, it has its own genes encoding for a separate one, they don't have these translocation domains. They don't have chaperones. But what they have is a conserved N-terminal domain amongst all of them that essentially functions in its own way to target, but independently of the chaperones. So yeah, it shouldn't be understood that the chaperone signal is the only way that targeting can be done. It's just one of the main ways, and to date, the only one that's been characterized in terms of what that signal might be. Margie? Oh.
Also, I guess about the beta motifs, is it clear, does the beta motif have anything to do with the specificity of certain substrates for their chaperones?
Well, that's an interesting question because there are several classes of these types of chaperones. There are sort of the monogamous ones that there seems to be a secretion chaperone that pairs up uniquely with one substrate. And then there are more promiscuous ones. And in fact, the one that I was trying to illustrate there was CYP-A, the INF-B chaperone, binds several different effectors. What you find in this interaction is a lot of slop, actually. So, and I can even illustrate that with one of the monogamous chaperones. So these chaperones form homodimers. And the pocket that binds the beta motif is on opposite sides. So there's one of these from Salmonella, more alphabet soup, but it's called SPTP. The translocation domain runs around the homodimer of chaperones, interacting with one of these chaperone pockets, and then hits the other chaperone pocket. It has two beta motifs to confer that interaction. Those are not conserved. They're conserved in that the hydrophobic nature, the positions of those residues are there, are very similar, but the actual, it's not identical in terms of the amino acids involved. And that's basically what you see across them, is that there's a lot of slop in this interaction, which is not that surprising. You have a large hydrophobic pocket, and basically you're sticking in a hydrophobic region. So there are two, and it's not just that. So the strand itself is partially targeted by making the intermolecular beta sheet, and then it's also anchored by the beta motif residues that plug into the pocket. So there's some targeting of that region, but then there's also a lot of slop. We've tried to do some things where we tried to turn one for more specificity into the other, but we clearly don't understand what we're doing with that because we haven't been able to achieve that in any sort of predictive fashion.
The H. pylori tag protein, its interaction with ASC, is that the same surface that
Interacts with the kinase?
Yeah.
No, it's in two very different regions. So the kinase interacts with a far C-terminal region of the protein that up until very recently was kind of the business end of the molecule, but mainly because that's where everybody had studied. So it had that region has an important tyrosine phosphorylation motif that recruits a lot of eukaryotic signaling factors, and that's one of the ways it interacts with a lot of proteins. So one of the reasons we went after these subdomains at the N-terminus, so 800 amino acids south of that domain that binds the kinase, is because so little was known. And so we, at the time we began work there, it wasn't even clear what the domain structure of that was. It was just a piece of structure that was very poorly behaved in most biochemistry. So we worked on that for a couple of years, and we identified these separate domains, one of which was the ASP2 binding domain, but that's a good distance from the C-terminal domain. Now, whether or not that all folds up together into something more complicated is an interesting question, but we don't know the answer to that. Thank you.